All right, everyone, get ready and buckle up. We're about to go on a roller coaster through transcription. The central dogma of biology is where DNA is transcribed into RNA and RNA is translated into protein. In this video, we will solely be focusing on the transcription in prokaryotes. Some information that is important to know is that DNA is always used as a template, and RNA grows 5' to 3'. RNTPs are used and have an OH at the 2' position, and uracil is used instead of thymine. These are shown in the red molecule in the bottom right. The blue molecule is the 3' end of the RNA. If we cleave off 2 phosphates from the RNTP, a phosphodiester bond is formed between the leftover phosphate, an incoming nucleotide, and the OH on the 3' end of the RNA strand. I would like to begin talking about identifying the coding strand and the template strand. Since we have transcription going to the right and RNA is being transcribed from 5' to 3', it will look like this. Next, the top strand will be the coding strand because it is identical to the RNA sequence, and the bottom strand will be the template strand because it is complementary to the RNA strand. This is the strand in which nucleotides are built off of. The promoter region contains several sequences that allow RNA polymerase to bind. The positive 1 nucleotide seen here will be the first one that we transcribe. This is the starting place for transcription. If we move upstream, more to the promoter region, we will end up at the -10 consensus sequence, which is very important in allowing RNA polymerase to bind during this process. Then, if we move upstream a little more, we will go to the -35 consensus sequence. This is also involved in RNA polymerase binding. RNA polymerase makes phosphodiester bonds in the 5' to 3' direction. The sigma factor subunit is part of the hollow enzyme, and it helps find the promoter in sending the RNA polymerase to the start site of transcription. The sigma factor then has to be removed in order for RNA polymerase to begin moving down the chain. Next, we will discuss the RNA coding region. This is continuously transcribed and is downstream on the transcription roller coaster. In prokaryotes, once this gets to the terminator, transcription will stop. Now we will dive into this a little more. When we are in termination, if an inverted repeat occurs, this is called independent termination, and there is a hair loop that can be formed. This is part of the roller coaster that is a loop-de-loop, which might make you riders feel a bit sick. This hairpin loop causes RNA polymerase to pause, and there is a poly A tail at the end where they just code for use, helping to dissociate the RNA from the DNA, thus ending transcription. If the -10 box, or the TATA box, is deleted in a gene that codes for an enzyme to metabolize sugar, then there will be a loss in the sites that code for this, causing there to be an insertion of extra base pairs to make up for this deletion. This can create a potentially different enzyme being formed, and it will most likely code for something different than metabolizing sugar. Alright, it seems as if this roller coaster has come to an end. Thanks for tagging along this ride all throughout transcription in prokaryotes.